Hello everyone. In this video lecture, we are going to study about immunity. It is the first lecture of immunology, so let us discuss about immunity. Now, what is immunity? I would not mention any definition here, but I would suggest a method to recognize what is immunity. So, immunity is the capacity. Immunity is the capacity of the body. It is the capacity of the body to resist pathogens. So, what is immunity? Immunity is the capacity, warm capacity, body's capacity to do what? To resist warm pathogens which are entering in the body. So, immunity is the capacity of the body to resist pathogens entering in the body, and these pathogens can be anything: virus, bacteria, anything. So this was the definition of immunity, that it is the capacity of the body to resist pathogens which are entering in the human body. Now, immunity is further divided. So if I will talk about immunity, immunity is divided into two subtypes. The first one is innate immunity. And the other one is acquired immunity. Now, what is this innate immunity and what is acquired immunity? Now, innate immunity is the capacity, same capacity of body to do what? To resist warm pathogens. But this innate immunity is inborn. So it is the capacity, but it is the inborn capacity. Similarly, acquired immunity is also the capacity, but that capacity is the acquired capacity. Wow. You should also say wow, because here we are learning the real feel. We are getting the real feel of these terms. Innate immunity and acquired immunity both are the capacities of the body to resist pathogens entering in the body. But innate immunity is inborn capacity. It is present from the birth. But in acquired immunity, it is the capacity of the body to resist pathogens. But it is the acquired capacity. Okay. Now, if I will talk about innate immunity is non-specific. Now, how we can say that it is, why innate immunity is known as non-specific? Generally, it is not mentioned in the book, but as you have followed Enabler channel, I should mention it. Innate immunity is known as non-specific because it respond all pathogens. It respond all pathogens, or it work against all pathogens in the same way. Whether it would be virus, whether it would be bacteria of any strain, of any type, it would work similarly against all the pathogens. That's why it is known as non-specific. It will not work specifically to a particular antigen. But acquired immunity is specific type of immunity. Now, why I am saying it is specific? Because it will work only against specific antigens. Against specific pathogens. If a particular organism has acquired the capacity to resist the pathogens during its life cycle, that would be specific. That's why it comes under acquired immunity. Now, it has various examples, like we give vaccine to an organism. So that vaccine works only for a particular strain of bacteria or a particular strain of pathogen. It doesn't work for all in the same way. That's why acquired immunity is known as specific and innate immunity is known as non-specific, because innate immunity works or responds against pathogens in the same way. So this was about the innate and acquired immunity. Now this acquired immunity is further divided as cellular immunity, and humoral immunity. Now, what this cellular and humoral immunity means, we will study this in detail in the upcoming lectures, but we have to mention here that cellular immunity is maintained by the T lymphocytes and the humoral immunity is maintained by the B lymphocytes. Now, in this video lecture, we will focus mainly on the development of the T lymphocytes, how T lymphocyte develops and they become active in the blood of the organism. So let us discuss it. As we all know that we have certain cells in the bone marrow. You know bone marrow? Bone marrow is present in the medullary cavity of long bones, bone marrow. This bone marrow has a cell known as pluripotent stem cell or pluripotent hematopoietic stem cell. This pluripotent stem cell give rise to two types of cell. One is lymphoid progenitor cell and other is myeloid progenitor cell. Actually, this pluripotent cell is very important cell because this cell will give rise to all constituent of blood, as T lymphocytes are also the constituent of blood, as we know, because where lymphocyte would be present, how they will travel to different sites via blood. So this lymphoid progenitor cell will produce three things: T lymphocytes, B lymphocytes and natural killer cells. Okay, and all other constituent would be given by the myeloid stem cell. Okay, now in this video lecture we will generally focus on the T lymphocytes. So T lymphocytes are firstly produced by the bone marrow. Where they are produced? From where they are produced? They are produced from the bone marrow. Now when they are produced from the bone marrow, they get into the thymus gland. They move towards the thymus gland via blood. Why they move to the thymus gland? They move thymus gland to get processed, to become active cells. And until activation, until being functional, they are known as thymocytes. What they are known as? Thymocytes, isn't it? Now, what happens in thymus? How T lymphocytes only get into thymus? Why they don't get anywhere else? Because as a doctor, as a scientist, we have to be reasonable. We have to question ourselves in each and every portion that why it happens. Because it's the basic funda of science to question. Now it gets into thymus only because thymus releases chemokines. Chemokines are certain chemicals which will attract these thymocytes in its cortex. Now why they are attracting this thymocytes? Because when T lymphocytes are produced from bone marrow, they lack two important molecules, and those molecules are CD4 and CD8. These molecules are very important for which thing? To recognize the antigens, the pathogens. So these CD4 and CD8 molecules are absent in the T lymphocytes when it is released from bone marrow. That's why at that particular stage, the lymphocyte which lacks CD4 and CD8 is known as double negative T lymphocyte. Which lymphocyte? Double negative. Okay. Now what happens in the cortex? There's these chemokines. What they do? They stimulate these T cells to produce CD4 and CD8 molecules on the surface of T lymphocytes. And after producing the CD4 and CD8 molecules on the surface of T lymphocyte, they are known as double positives. Now here a term comes. If the T lymphocyte is not able to bind with the CD4 or CD8, let it be first. Let us come to the basics first, then I will discuss it. Now what is the importance of this CD4 and CD8? See, there is another molecule present on the antibodies or another cells known as MHC1 and MHC2. Just keep it simple. This is the T lymphocyte with CD4 and CD8. These are the important molecules for T lymphocyte so that it can be functional, it can process well. Now, this CD4 will bind to MHC2 molecule and this CD8 will bind to MHC1. Now, if T lymphocyte fails to bind with MHC molecules, then it is known as, then apoptosis happens. Which thing happens? Apoptosis. That is destruction of these T lymphocytes. Because the CD4 and CD8 will not be able to bind with the MHC1 and MHC2 molecule, which is very essential for them to being functional. If CD4 and CD8 MHC2 MHC1, see, their binding is very important for T lymphocytes to be functional. If this particular thing doesn't happen, then these will go under apoptosis, destruction, self-destruction. So this is known as positive selection. Why it is known as positive selection? Because those cells which will not bind with MHC1 and MHC2, they are non-functional. So thymus has to remove them. So that selection is known as positive selection. Now, after getting selected, they are transferred to the medulla of thymus gland. Here, these T cells are exposed to self antigens like self cells of the body. Now, tell me, should these T lymphocyte kill the cells of own body? No. T lymphocytes should kill the cells of foreign body, not the self body. So those T cells which will recognize or which will kill or which will just bind with the self antigens, they are undergone apoptosis. That means they are destructed because they could be harmful, they could be dangerous for the body. That's why they go under apoptosis. Which cell goes apoptosis? Those T cells which will bind to the self antigens, they go under apoptosis. After it, now this particular thing is known as negative selection. So these selections are for what? To kill the T lymphocytes which are harmful for the body. First, if CD4 and CD8 can't bind to MHC2 and MHC1 simultaneously, then they are undergone apoptosis. If these T lymphocytes will bind to self antigen, at that time they are also undergone apoptosis. The first is known as positive selection and the next one is known as negative selection. So simple. I just revised it in a jiffy for you. Now there is important thing at your disposal now. Now these T cells, T lymphocytes are exposed to body cells now. Now if these T cells will bind to MHC1, now if, as we all know, the T cells will have CD4 and CD8 molecules on the surface. If MHC1 will bind with CD4 molecule, then the function of or CD8 will down regulate, that its function will be down regulated. That means only CD4 would be functional if it is binding with MHC2. So it would be known as helper T cell. But if MHC1 will bind with CD8 and the function of CD4 would be down regulated, then this would be known as cytotoxic T cell, and they are finally released in the blood for their function. So see how beautiful is this process, and I made it clear for you that how, like I will just repeat it again for you, that CD4 and CD8, both the molecules would be present on the cell surface, T cell surface. But if among CD4 and CD8, if CD4 bind to MHC2, what will happen? The function of CD8 would be down regulated, and that T cell would become helper T cell. In the other case, if an MHC1 will bind to the CD8, the function of CD4 would be down regulated and the cell would become cytotoxic C cell. So this was the first lecture of immunity. In the upcoming lectures, we will deal with something new. So thanks for your cooperation.